So here's the last bit of information from Chapter 12. Sorry that some of this stuff is a little bit out of order. Our current textbook, although it covers the same exact stuff my other textbook, Principles of Life, covered, a few things are in different orders. You can find them all. And then our current textbook, Campbell, adds this information in now. We'll probably revisit it again when we talk about mutations and what happens in the genome. But since these mutations are the result of big chromosomal changes, it's appropriate to talk about it now. So in meiosis, remember, it's very important to move the chromosomes. In meiosis 1, the sister chromatids stay together. In meiosis 2, the sister chromatids separate. If at either of these places we have what's called non-disjunction, the appropriate chromosomes do not separate, you got a big problem. You're going to have it that you have the wrong number of chromosomes in your haploid cell. So now normally during reproduction, if a sperm that perhaps has the wrong number of chromosomes meets up with an egg with the correct number of chromosomes, right after fertilization, a big matching event happens to make sure that you have the right number of chromosomes. Normally, our bodies cannot handle the wrong number of chromosomes, and the cells will abort, probably go into an apoptosis state, and that embryo dies. There's a few examples that can survive, and the big one is Down syndrome. Trisomy 21 is non-disjunction of chromosome 21, and humans can handle 3 copies of that. We have the whole plethora of effects of being a Down syndrome person, but it does not result in death. In fact, those people can live a pretty decently long life. They just have heart defects as well as the facial features and depressed cognitive skills. So, sorry about those planes going overhead. So there's some other things that can happen wholesale to a chromosome when it's getting ready to be moved around. So you can have a deletion event take place. So for example, they're just giving you a code on this chromosome. If this hunk for some reason gets chopped out, maybe crossing over didn't happen correctly, you'll now be missing it. Deletion, pretty obvious. Duplication. Again, probably the result of a crossing over abnormality where a chunk gets put in twice on one chromosome. It's probably going to be missing from the other chromosome. And now you have 2 copies of that genomic information. Again, most of the times our bodies can't handle having more than what we're supposed to have of a gene. We're going to discuss that as we keep going through the genetic section of the textbook. And this might result in a disease state or death. Inversion. Again, probably a crossing over issue where now a whole hunk of the chromosome, B, C, D, they have labeled here, gets reinserted the wrong direction. Imagine if there was a critical base pair sequence here between A and B. We now just disrupted that completely. We could have all kinds of big problems. And then translocation. This sounds like a huge ordeal that we're taking a hunk of one chromosome, so AB, ends up on this chromosome and vice versa. Believe it or not, it happens at a few breakpoints in some chromosomes. One's called the Philadelphia chromosome. It's a common signaling defect and results of in a tumor cell cancer. Oops, that's all the same stuff I just talked about. So this actually happens more than you think, or at least we can notice a few. And there's a very famous one where this sequence here, that sequence there is disruptive and makes a activated gene that tells a cell to go through the cell cycle. So that's the end of the chapter. Hooray! Get ready for the test.